Good day, my name is SURESH Ramasamy. So this is about the lab experiment we did for IEA514 entitled Toxic Cyanobacteria Detection Study, because basically we want to show how we run the experiment to identify this bacteria. The experiment demonstrates a molecular workflow involving DNA extraction, PCR amplification, gel electrophoresis, and sequence identification using NCBI blast. Now, for the lab setup and sample preparation, the procedure began with proper laboratory preparation to minimize contamination. The workbench was cleaned with 70% ethanol and gloves were worn throughout DNA handling. The samples were transferred into microcentrifuge tube and centrifuged to concentrate the microbial cells. After centrifugation, the supernatant was removed carefully, leaving the cell palette for DNA extraction. DNA extraction was carried out according to the bacterial DNA extraction protocol. First, buffer R1 was added to resuspend the cell palette completely. This is important because incomplete resuspension can reduce lysis efficiency. Lysozyme was then added to digest the bacterial cell wall, followed by incubation. After that, the cells were treated with buffer R2 and proteinase K to lyse the cells and digest proteins that may interfere with DNA recovery. Buffer Bg and absolute ethanol were then added to support DNA binding into the spin column membrane. The mixture was transferred into the spin column and centrifuge. The column was washed to remove impurities, salts, proteins, and possible PCR inhibitors. Finally, preheated elution buffer was added onto the membrane to recover purified DNA for PCR analysis. Now, for PCR preparation, the reaction mixture contained DNA template, MyTech read mix, primers, and sterile distilled water. The MyTech read mix provides the DNA polymerase, nucleotides, and buffer system needed for amplification. The primers target the cyanobacterial 16S ribosomal RNA gene, which is commonly used for bacterial and cyanobacterial identification. The tubes were prepared carefully to avoid cross-contamination before being placed into the PCR machine. Now, for the PCR amplification and gel electrophoresis, in the PCR machine, the DNA underwent repeated cycles of denaturation, annealing, and extension. These cycles amplified the target DNA region to the detectable quantity. The PCR product was then analyzed by agarose gel electrophoresis. Since DNA is negatively charged, it migrated towards the positive electrode. The presence of DNA band indicates successful amplification of the target sequence. Now, the amplified sequence was analyzed using NCBI blast. The result identified the sequence as a partial 16S ribosomal RNA gene. The best match showed about 98% coding coverage, 97.31% in identity, and an e-value of 0. The closest named cyanobacterial matches were related to Nodoxyllinia and Leptolimpigia. Therefore, the sequence supports cyanobacterial identification at genus or lineage level. But it does not confirm toxin production because no mice eat toxin gene was identified. Thank you very much.